today to be able to introduce our third Royal, Dr. David Anderson, the Seymour Benzer Professor of Biology at Caltech, and as I said, the third Barondes lecturer. Dr. Anderson received his A.B. from Harvard, and then his PhD in cell biology at Rockefeller University, working with a Nobel laureate, Hunter Blavel. He then moved on from one Nobel laureate to another, doing his postdoctoral training at Columbia with Richard Axel. He's had a remarkable career. I had a chance last night to sit down and take a look. This is one of the fun things about doing these introductions, and really to see someone who's made fundamental contributions, not just in a single area of science, but really in multiple areas of science. So many of you may not know, if you're familiar with his current work, that really for the first epoch in his career, the first 20 years, his research focused on the biology of neurostem cells. He was the first to isolate a multipotent stem cell from a mammalian nervous system, and subsequently then identified both growth factors and master transcriptional regulators that control their differentiation into neurons and glia. He made the unexpected and fundamental discovery that arteries and veins are genetically distinct, even before the heart begins to beat. And he combined his interest in discovery and neural development with this insight to contribute to an expanding and incredibly important field in the intersection and the molecular crosstalk between developing nerves and vessels. But then, if that wasn't enough, for an encore starting about 15 years ago, he shifted his focus from neural development to the study of neural circuits that control emotions and behavior. He's employed a remarkable range of technologies, being a leader in using optogenetics, pharmacogenetics, electrophysiology, in vivo imaging, quantitative behavioral analysis, and machine vision-based approaches, and developed and applied these tools to identify and manipulate cells and circuits involved in the emotional behaviors in mice. And if that weren't enough, he has now mounted a parallel inquiry that dissects the genes and circuits underlying behavior in fruit fly. And he's become, I would say, the leader internationally in this field. Now, sometimes people wonder what can the fruit fly, well, actually this audience I hope doesn't, but wonder what the fruit fly can tell us about something as complex as neuropsychiatric disorders. But the kinds of internal states that he's studying really form the evolutionary underpinnings of emotional behavior in higher organisms, and I think offer a remarkable opportunity and unique avenues to illuminate the biological basis of the most mysterious of human maladies. So at Caltech, Dr. Anderson is the Tianqiao and Chrissy Chen Leadership Chair and Director of the Tianqiao and Chrissy Chen Institute for Neuroscience. To the envy of the vast majority of his scientific colleagues, he's been an investigator at the Howard Hughes Medical Institute since 1989. I certainly envy that. Dr. Anderson's awards are too numerous to list without taking up the rest of the lecture time, but I'll just say that he gets to add to a host of recent awards and now the Barondes Lecturer. He's a fellow of the American Academy of Arts and Sciences, and in 2007 was elected to the National Academy of Science. So today he's going to talk to us about neural circuits, controlling innate social and defensive behaviors, and once again we're incredibly grateful and thankful that you have agreed to come and join us.
Thanks, Matt. It's really a pleasure and an honor to be the Barondes Lecturer. I was thrilled to be invited to do this, not only because Sam is a role model in his Menschlichkeit and in his understanding of how basic science relates to psychiatry, which is something that I care deeply about, but also that he's somebody that I have admired throughout my career. And so I really appreciate that, and also the fact that this is at UCSF, where I have so many friends and so much history. Before I start, I want to dedicate this lecture to Alison Dope. Many of you knew Alison. Many of you may not know that Alison was an extremely close friend of mine. I based my postdoctoral research on discoveries that she made while she was an MD PhD student at Harvard, and she moved to Caltech to be a postdoc in Mark Kanishi's lab just as I was starting my own lab there in 1986, and we became very close to each other and stayed in touch for many years, and it was really tragic to lose her, but I know that Sam was a very important mentor of Allison, and I just wanted to acknowledge the influence that she had on my life and in my science. So, as I said, Sam has been sort of a guide, and speaking of guides, it was Passover recently. Many of you may not realize I celebrate Passover because of my last name, but actually I do. And Sam might be interested to know that in honor of current events, we adopted a recent version of the Haggadah, which is the guide that you use to take you through the Seder. And this is the Attorney General's version of the Haggadah. So for all of you who are thinking about what to do next year for your Haggadah, I thought I would show you this. And as you'll see, this has influenced me now in thinking about not only the Seder, but also science as well. So let's get back to the topic, which is the relationship between emotion and psychiatric disorders. I think most people would agree that psychiatric disorders are fundamentally disorders of emotion systems in the brain. And the first to recognize this actually was Charles Darwin, who's the author of what, in my opinion, is the first serious attempt to try to think about emotion from an evolutionary standpoint, the function that emotion subserves, and why emotional expressions appear to be conserved in evolution. And Darwin, in his monograph, already noted the relationship of emotion to psychiatric disorders. He had this image of a patient taken from a mental institution and entitled it "Photograph of an Insane Woman to Show the Condition of Her Hair." So while this is, from our distant perspective, this is quaint and politically incorrect and scientifically incorrect, it also reflects the fact that Darwin viewed emotions as internal states that are expressed by particular behaviors, expressions, and also the state of the body and all of its associated expressions. And so just to say that Darwin realized that emotion and mental illness were deeply connected to each other. How that is and what emotions are remains a matter of great debate to this day. In fact, I think it's safe to say that in the field of emotion, unlike many other fields such as cell biology, genetics, molecular biology, developmental biology, there isn't even a consensus on what emotions are or where we can study them. And in fact, one of the central debates that has been around since the time of William James and Darwin, and that has reemerged in the last few years, has been the issue of whether we can even study emotions in animals as well as in humans. There are many people that would believe that emotions are a property, at least of mammalian organisms, but it's not clear whether that attribution, which is after all just based on anthropomorphization, extends to more distant organisms, including invertebrates. And most of what we have to guide us is a matter of opinion. Darwin was an unabashed anthropomorphist in this respect. He simply flat out believed that all animals had emotions and emotion states. Even insects, he wrote, express anger, terror, jealousy, and love by their stridulation, the moving of their wings. On the other hand, another one of my heroes, Nico Tinbergen, the Dutch ethologist, wrote that hunger, like anger, fear, and so forth, is a phenomenon that can be known only by introspection. When applied to another species, meaning anything other than humans, it is merely a guess about the possible nature of the animal's subjective state. And so we have on the one hand the idea that the essence of emotion is subjective experience. We can only know whether there is subjective experience in an animal that's capable of self-report, which restricts it to humans. Therefore, by that definition, emotion can only be studied in humans. And then we have the idea of Darwin that emotions are some sort of internal state that can be expressed by measurable behaviors and other features. And so my colleague Ralph Adolphs and I at Caltech have tried to take this on, and our view is really an extension of an idea that Kent Berich articulated in his thinking about motivational states. and that is that emotions are a class of internal states that you can think of as intervening variables in between that link a series of different types of stimuli from outside the organism, inside the organism, to a series of responses which include not only motor behavior, somatic responses, but also in human subjective feelings. And the idea here is that subjective feelings are not the defining feature of emotions. Rather, subjective feelings are but one of many readouts of emotions. They're one that we can read out in humans and not in animals, but there are many other readouts of emotion states that we can study in animals as well as in humans. And so our approach to this has been to try to think about and understand what are the defining features of these central states and how can we study them. And we've asked the question whether there are general properties that are common to different emotion states. And some of these properties, which we refer to as emotion primitives, you can think of them as sort of evolutionary building blocks of emotion that distinguish emotional responses from pure reflexes, are scalability, the idea that emotions are graded in their intensity and the behaviors that express them can be graded in their intensity and they can change at different thresholds, that they show persistence, that emotional responses and emotional behaviors outlast the stimuli that triggered them, unlike reflex responses, which are generally time-locked, that emotions have a valence, they are positive and negative, and that they are generalizable. An emotion triggered under one circumstances can apply to behavior in another set of circumstances. You get into a fight at a bar, one of those Irish bars Peter used to take me in San Francisco, and then you come home and your kid is screaming and you get mad at your kid. And so the question is, can we begin to try to understand how these emotion primitives are implemented by brains? What are the underlying mechanisms? Not that there has to be a unitary mechanism for each of these. And importantly, can they help explain the evolution of emotion as a neural control mechanism, a class of neural control mechanism? So that's what I want to focus on in this talk. Particularly what we've learned about persistent internal states. This is my Attorney General approved outline for the talk. One is the control of I'll tell you a bit about the control of a predator-evoked internal defensive redacted, and control of a persistent internal state of social redacted, which we've studied in flies. And then finally, control of aggression and persistent fear by social redacted, where we were able to link together our studies in flies and in mice. And I'll show this again after the congressional subpoena removed the redactions at the end of the talk. So to begin, I want to focus on an innate fear pathway. So as many of you know, animals have things that they are innately afraid of. Some of you may be innately afraid of tarantulas, even though you may never have seen a live tarantula. And there are also, of course, are things that we can learn to be afraid of, like a nasty black dog that bit us when we were a kid. And much of that work is focused on the role of the amygdala, a structure in the medial temporal lobe, to the extent that many people believe that the amygdala equates to fear. All that is fear is in the amygdala, and all that the amygdala does is fear. And today I want to tell you about part of the brain in another structure called the hypothalamus, which we think plays an important role in fear, but not of the learned kind, rather the type that is evolutionarily prepared, that is, where the animal shows a response in the absence of any previous exposure to the stimulus. And since we are reductionists at heart, thanks to my training with Peter in Günter Blobel's lab, we focus on particular populations of neurons in brain structures that give us genetic access to those cells. And we're focusing here on one particular nucleus in the hypothalamus that's called the ventromedial nucleus of the hypothalamus, and a subset of neurons in the dorsomedial region of that structure that are labeled in green by a transcription factor called SF1. These neurons are different from the neurons that I usually talk about, which I'll mention only briefly today, in the ventrolateral pole that control aggression, mating, and other social behaviors. These neurons can be accessed, as we do with other studies of this sort, using lines of transgenic mice that express Cre recombinase in these neurons. We didn't make them in this case, they were generated in Brad Lowell's laboratory, but as a result of that, we can manipulate these cells using light via optogenetics. We can activate them, we can inhibit them, we can image their activity and begin to learn something about their function in this behavior. Now, as we began to perturb and tinker with the function of these neurons, we noticed that activating these cells controlled and produced defensive behaviors like freezing, but not in a fixed unitary way. Interestingly, what we found is that we were able to evoke different kinds of defensive behaviors at different intensities of optogenetic stimulation, so that the weakest intensity of stimulation produced an avoidance reaction where the animal would move away from the side of a box where the stimulation was being delivered. Slightly more intense stimulation caused the animal to stop moving altogether and freeze, and even stronger activity stimulation would cause the animal to transition from freezing to undirected escape, where it would run around the cage and try to leap out of the cage. And so this attracted our interest because, at least phenomenologically, it seems to reflect this property of scalability, that activating these neurons controls different kinds of defensive behavior in a graded and threshold-dependent manner, as if there is some sort of a rheostat-like control operating the decision to freeze or to flee. In addition, what we found is that these neurons are capable of producing persistent responses, defensive responses. And if you think about it, persistence is really one of the hallmarks of emotional responses. If you're walking along a trail and you hear the rattle of a snake next to you, you'll jump up into the air and long after the snake has slithered off into the bushes, your heart will continue to pound and you'll be hypersensitized such that if you see something, a stick that even looks like a snake, you're likely to jump. And so that raises a number of important questions that I want to focus on, whether there are neurons that trigger persistent defensive behaviors, whether they themselves are persistently activated by threatening stimuli, what the function of this persistence is, and how this persistence is implemented at the circuit level. And these are the questions that I would like to proceed with now in this section of the talk. So as I said, we discovered that these SF1 neurons in VNH, when activated, control defensive behaviors in a graded, scalable way, but they also produce persistent behaviors. So you see in this same real-time place avoidance assay that when the mice are out of the side of the box where the optogenetic stimulation was delivered, they run around the perimeter exhibiting a behavior called thigmotaxis, in contrast to controls, which is a typical sign of anxiety state in animals, and this can last for many minutes. Furthermore, the animals show persistent freezing. I'm sorry that the contrast is a little bit low here, but after the offset of the optogenetic stimulation in blue, after the activity burst, there's no longer any optogenetic stimulation, but the animals are freezing here for many, many seconds, as you can see down below. And so these and here's another example of this persistence. When you activate these neurons, it interrupts any kind of ongoing appetitive behavior. Mating is interrupted, feeding is interrupted, and interestingly, once you turn off the light and ask how long until the animals reinitiate whatever they were doing when you stimulated the neurons and interrupted the behavior, in those animals where the behavior was interrupted by stimulating these cells, it takes them longer to get back to doing whatever they were doing. So here the animals were mating. When we activated the cells, they stopped mating very quickly in comparison to controls, and it took them significantly longer to reinitiate mating after we stopped stimulating the cells. So that suggests that there's some sort of persistent internal state that is being triggered by activating these cells, which is outlasting the stimulus and which has to dissipate in order for the mating to be reinitiated. So altogether, these data suggested that this population of neurons, at least when artificially manipulated, produces behaviors that have many of these emotion primitive features: scalability, persistence, valence, generalization. I didn't show you this. And they also cause an increase in stress hormones, heart rate, and pupillary dilation. And all of this in the absence of any experiments to show that these cells are capable of causing or allowing conditioning of a fear response. In fact, if you try to use these neurons as an unconditional stimulus, like a foot shock in a freezing experiment, it doesn't work. And I should say that similar data were reported by my former postdoc, Dayu Lin, who now has her own lab at NYU. So I want to drill down a little bit more now and tell you about some unpublished work regarding the mechanism and implementation of persistence. And the first critical question is, are these neurons themselves activated by threatening stimuli in a persistent manner? And so initially we looked at this by carrying out bulk calcium imaging. This is a technique many of you have heard of called fiber photometry. Here we express GCAMP, which is a calcium-sensitive version of GFP. In these neurons, we put a fiber photometry, optic fiber in the animal's brain, stimulate it with an excitation beam, and then we just look at the overall level of activity, which is measured here. And now we're introducing the stimulus, which is a rat. I apologize for the crude nature of the stimulus presentation, but this experiment was done by a molecular biologist, not by a systems neuroscientist, so it lacks the sophisticated servo-controlled closed loop. But what I want you to see is that even after the stimulus is removed, the level of activity remains very high inside this animal's brain among these neurons in this region. And it's difficult to quantify the type of behavior that the animal is showing, but if you have a lot of experience watching mice, you can tell that it's showing a sort of tentative behavior moving around the cage. And so we can quantify this and show that we get this type of persistent response when we use a predator, a rat, as a stimulus, but not if we use a toy rat. And also that the decay constant, that is the duration of the persistence, is longer using a rat than if we use a mouse, a conspecific. So I don't want to say that this is a fearometer or anything like that, but it is an interesting correlation that suggests at least that exposure of the animal to this type of stimulus could produce a persistent response. Now, many of you will immediately say, well, how do you know that this is really a persistent response, that the rat didn't just pee in the cage, and this isn't persistence of the odor of rat urine? And I'll show you some additional evidence for that shortly. But the first thing I want to address is whether this persistent activity has any behavioral function. And so it's been challenging for us to try to answer that question, but recently we've made some progress on this, and this is Lingyun Li, a postdoctoral fellow in the lab, who developed an assay in which we expose a mouse which is in a large open arena to a rat, not held as shown here in the diagram as a naked rat, but in a cage with diapers to catch any droppings or drippings for only 15 seconds. And that 15 seconds of exposure to the rat is enough to switch the mouse from moving randomly around the cage to hugging the walls in this thigmotaxis open field behavior. And so the experiment that we tried to do was to ask what happens if we silence these neurons in VMH during the persistent phase of the thigmotaxis after the rat is no longer there. And we used a variant of an optogenetic approach to silencing these neurons where blue light opens a chloride conductance channel to hyperpolarize the cells. And so the design of the experiment looks like this. We have a positive control with one set of animals where silencing is initiated before the rat is even presented to the mice. And then we have another experiment where the silencing is only initiated after the rat has been presented and is removed. And what you can see here, and perhaps most clearly here in the graph, is that there is a substantial reduction, first of all, in the positive control. The thigmotaxis behavior is strongly suppressed, indicating that the activity of these neurons is indeed required for the animal to show this sort of wall-hugging behavior in response to the rat. But interestingly, you see that strong suppressive effect kick in even when you deliver the stimulus after the rat has been taken away and the mouse is just walking around by itself. And you see again a strong and significant suppression of behavior. That doesn't mean that this is the only process that is contributing to the persistent behavior of the rat, the persistent of the mouse, the persistent movement, but it does argue that this persistent activity that we can measure does play a role in controlling this type of persistent defensive behavior in the animal. Then the next question becomes, how exactly is this persistent activity represented in this collection of cells in this nucleus, which is on the order of about 4,000 neurons, just to give you a rough idea, maybe 4,000 to 5,000 neurons on each side of the brain. And there are two extreme possibilities that you could imagine. One is that all of the neurons are synchronously activated by the rat, and all of the neurons show a synchronous persistent activity that lasts in parallel. The other possibility is that the population persistence that we see when we just measure bulk activity from these cells, in fact, reflects is a population phenomenon. It reflects the superposition of many individual neuronal responses, each of which has a slightly different time to peak and different decay. And so the way that we asked that question was now to carry out imaging of individual neurons deep in this brain region using the Inscopics miniscopes developed by Mark Schnitzer and his colleagues at Stanford. So we put a 6-millimeter long glass lens deep into the brain above the neurons expressing GCAMP in this region and clamp a 2-gram microscope to the animal's head. And then in this case, we developed a head-fixed preparation where we could expose the animal to the predator in a time-locked way and ask what neurons are activated and in what kind of pattern. And the results, as you can show here, are very clear. Each of these traces is from a different neuron, and there are a pair of responses shown from a pair of trials for each of these neurons. And as you can see here from the black dots that mark the peaks for each of these cells, they're not all peaking at exactly the same time, but rather there is a heterogeneous set of responses that initiate and peak at different times and show slightly different times of decay. So that suggests to us that this persistence in the structure as a whole is a population phenomenon rather than reflecting synchronous activity of all of the cells. Now you may still be wondering, well, how do you know this is really a persistent response? How do you know there is not some lingering ode to rat that is left behind even when the rat is presented in a cage like that? And we worried about this as well, and so we asked whether we could find an alternative modality of threatening stimulus where we could have better temporal control of stimulus onset and stimulus offset than we can using an intact animal. And here we used patterned ultrasound, and so we had shown back in 2003 in one of the first papers we published as we got into this new area that mice will exhibit defensive responses, including freezing or running, in response to ultrasound in the frequency range from 17 to 20 or 22 kilohertz. That's normally the range in which rats emit alarm vocalizations. This strain of mice doesn't emit such vocalizations, but nevertheless it is sensitive in that range. And so we asked the question of whether we would see any activity in these SF1 neurons in response to an ultrasound. And this was really an open question because the field generally views this hypothalamic nucleus as a sensory relay for olfactory cues, not for auditory cues. But to our surprise, this ultrasonic tone, but not a tone in the 2 kilohertz range, evoked a strong response shown here in purple with similar magnitude and similar slow decay kinetics as we saw from a rat. The interesting thing is that largely non-overlapping populations of neurons in this structure are activated by the rat and activated by the ultrasound, even though they both show this persistent pattern of activation. Indeed, the amount of overlap that we see between rat and ultrasound activated cells, shown in white, is what would be expected by chance. Now, Ann Kennedy, a computational postdoctoral fellow in the lab, has done some analysis using principal component analysis and various rotations of axes in principal component space, and has separated out this response into two components. Along one axis, the responses are well separated according to the modality of the stimulus. Here are all the rat responses over here, and here are all of the ultrasound responses over there. But at the same time, there is also a strong effect that both classes of stimuli show with respect to the intensity or salience of the stimulus. That is, the animals always show a stronger response to the rat and the ultrasound the first time they're exposed to it, and that habituates very quickly. So it suggests that perhaps in this region there is some sort of a mapping both of stimulus quality or stimulus identity and also of stimulus intensity. I apologize here, the legend I just realized is screwed up. This is this should be the rat over here in blue and the mouse, and then that's the tone. So Anne, because she's a computational neuroscientist, has been interested in the types of mechanisms that could give rise to this type of persistent activity at the population level. And I should say that when I describe persistent activity to a molecular biologist, their immediate reaction is, oh, well, there must be a neuropeptide or serotonin or a neuromodulator that's released, and that's what causes the persistence as it decays. If I tell a systems neuroscientist like Mike Stryker about this, they'll say, oh, there must be a recurrently connected excitatory network which is responsible for sustaining the activity. And indeed, this group of neurons is almost exclusively glutamatergic and is strongly recurrently connected. So Anne started building some computational models to see if she could produce the type of observed persistent responses with a simple recurrently connected network. And the answer is that she couldn't. The type of output from that simple recurrent circuit doesn't look anything like what we normally observe. All the units peak at the same time. She also considered the type of response that could be observed by having a sort of feed-forward series of inputs to this structure, and that also produced a very different type of response than what we observed. But if she added into a recurrently connected excitatory network a fast inhibition feedback loop with relatively slow excitation, then she produced a class of responses that are more similar to what we observe. Whether this is the actual mechanism, we don't know, but I should note that this nucleus is surrounded by a shell of inhibition, and so this certainly makes some predictions that inhibitory shell should be playing a role that controls the shape of the persistent response in this structure, and that's something that we can test. So the thinking that we have about these results at this point, although it's still very early days, is that we've identified a node in what's obviously a much larger interconnected circuit, a node that may integrate different types of sensory stimuli, auditory stimuli, olfactory stimuli, and compute in some way some kind of a scalar variable that may have something to do with the intensity or the salience of that stimulus, and then feed that out in some way to a downstream structure, which could be, for example, the periaqueductal gray, where these neurons are known to project, where there are thresholds that are set that interpret the amount of this output at different cutoff points, perhaps according to the level of inhibition that's being exerted on different downstream response circuits. This is purely speculation at this point, but it raises an interesting point that I wanted to make, which is that there may be a deep mechanistic relationship between these two properties of scalability and persistence, and that is, if you think about this node as a neural integrator, Studies of neural integrators, for example, the one that controls the oculomotor reflex that David Tank and Sebastian Sung has studied, which is this prototypical recurrently connected excitatory network. A critical feature of these integrators is that in order to be able to accumulate information about a changing stimulus with time, they have to have persistent activity. So the mechanism that allows for what I would call scalability here, the ability of this network to represent increasingly higher levels of a threat, for example, or the proximity of a predator, is dependent on persistent activity in this type of circuit, and that could be why, once the input has stopped, there is this slow and persistent decay. Again, this is purely speculation, but just to give you an idea of how we're thinking about this and that it does set up some interesting computational problems. It doesn't mean that there are no neuromodulators involved here. We've done single-cell RNA sequencing on these cells. There are many neuropeptides, many receptors, GPCRs, but I think the idea that a neural integrator could confer these properties of both scalability and persistence on certain behavioral responses is one that's at least worth considering. And one very preliminary piece of evidence that fits with this idea from some slice recordings that we have done indicates that there are some cells within this nucleus which, when optogenetically stimulated, can produce persistent firing lasting for many, many tens of seconds. So we intend to be exploring both the microcircuit mechanisms and also modulatory mechanisms underlying that. And before I leave this topic, I just want to emphasize, as I said earlier, that these neurons are immediately next door to, in neuroanatomical terms, a population of neurons marked by estrogen receptor and progesterone receptor, which Nira Shah has also studied in depth, and which we and he have shown control fighting behavior. So there's a sort of fight or flight axis laid out in different cell types along the dorsomedial ventrolateral pole of this nucleus. And in fact, we've shown with optogenetics that these ESR1 neurons control social behavior in this scalable, graded manner, with aggressive mounting and investigation produced at weak stimulation intensities, and attack being stimulated at higher intensities. And we've also observed persistent activity in this population of neurons in VMH triggered by optogenetic stimulation. So the idea here is that maybe there is a general principle here of how these two different populations of neurons control these innate defensive and social behaviors, and that's why they are located in this structure, which is almost entirely glutamatergic, as opposed to many of the other nuclei in the hypothalamus that have, if anything, more GABAergic than glutamatergic neurons. So there may be a principle here that emerges even while we're plowing through a lot of the details. And another concept that I'll return to in a second that I want to get across in both cases is the idea of being able to control certain kinds of behavioral decisions in this scalable or rheostat-like way. And what's interesting to me is that in both cases, the weak stimulation produces behaviors that you would consider to be low-risk behaviors, and only high-intensity stimulation produces higher-risk behaviors. So freezing is low-risk because it doesn't require much energy to freeze, and if the animal, the predator, doesn't see you, he's gone, whereas escape behavior is higher risk because you move and predators are highly motion sensitive, and so yes, you may run away, but there's a higher chance of being caught. The same thing between aggressive mounting and attack behavior, attack being much higher risk. And there are people that think about aggressive encounters from a game theory standpoint who have also invoked this idea that as these encounters proceed from the initial to the final stages, they go through low-risk types of behaviors to high-risk types of behaviors. So there may be something conceptually important in the ability of continuous control of behavioral outputs to move behavior from low-risk to high-risk as the intensity of activity in these structures is increased. And we have a long way to go to test this idea, but again, I just want to suggest that to give you an idea of how we're thinking about this. So to summarize, these SF1 neurons we've shown promote a persistent internal state of defensive arousal and threat alert. The Attorney General required that we redact the word fear. Here I've been very careful not to use the word fear because there's controversy about whether one should use that word in talking about animals. But if you want to, you should feel free to. These neurons exhibit persistent responses to threatening stimuli. The persistent activity of these neurons is necessary for some of these persistent defensive behaviors, and the duration of persistent activity, at least in this structure, is encoded at the population level. So I'd like to switch now and tell you about some of the work that we've done on persistent states using Drosophila as a model, and my eponymous colleague, the late Seymour Benzer, is the one who introduced Drosophila as a model to neurobiology for studying its genetics, and Drosophila exhibits a very well described courtship behavior as well as aggressive behavior. And an important question for us in approaching this system has been whether these behaviors are really purely robotic. The animal is just programmed by evolution so that when it gets a stimulus, it activates a pathway and the behavior is exhibited, and that's all she wrote, or whether flies have some sort of graded, persistent internal states that govern their behavioral responses when they are exposed to these stimuli, and if they do, how are these states encoded? And we've been guided in our thinking about this by the work of the neuroethology giants Nico Tinbergen and Konrad Lorenz. Tinbergen developed a hierarchical view of decisions between behaviors such as defensive behaviors versus reproductive, or mating and fighting, in which he envisioned that there would be specific nodes within the brain, nuclei, clusters of cells, which in the case of reproductive behaviors like mating and fighting were under the control of some higher level superordinate node that made the animal's brain go into the state of choosing between these alternative behaviors, and then that, for example, having chosen to fight rather than to mate or to nest, there would be further nodes that determined whether the animal was involved in threat behavior, biting behavior, or chasing behavior. And so he emphasized the binary control of these decisions, but he did propose that hormones or other internal state influences, if they existed, might exert their influence at the highest levels of this hierarchy. And then Lorenz famously proposed this hydraulic model of behavioral drive, in which he envisioned the brain metaphorically as a leaky bucket that contains something in it that can fill to varying levels to produce pressure that would evoke different behaviors, both according to the level of pressure and also to the valves, that is the degree of inhibition. And you can already start to see here a bit of an analogy to what I was describing earlier in terms of neural integrators converting scalable or graded output into different behaviors at different thresholds. So we focused on aggression in Drosophila. It's a complex social behavior. Both females and males show it. It's been described in ethological detail by Edward Kravitz and his colleagues. It involves a number of different stages and behaviors and escalation, including threat behavior, approach, boxing, fencing, and retreat. And this is an example of what you would consider a higher intensity aggressive behavior. Flies don't kill each other. They're more like sumo wrestlers. They just try to shove the other fly off of their territory. We decided to approach this like geneticists because basically I think like a geneticist. And so we decided to use the power of fly lines that express GAL4, the yeast transactivator, in different populations of neurons to carry out an unbiased and systematic screen to find neurons that functionally control different types of aggressive behavior. And this is a gain of function screen where we express a Drosophila trip channel in these neurons under the control of the GAL4 system. So the cells are off at 22 degrees and they're activated at 27 degrees. And then you can screen, the idea is to screen each of these lines for for increases in aggressive behavior. Now that's easier said than done, because if you want to screen thousands of these lines, which we did, you can't just sit there in front of a computer and do it manually, because it will take decades. And so we collaborated since the mid-2000s with Pietro Perona, a friend and machine vision specialist at Caltech, to develop automated methods based on machine learning and computer vision to track and automatically extract different types of aggressive behaviors from videos. And so we can multiplex this so that we can analyze 12 pairs of flies at a time. And Eric Hupfer, a postdoc in the laboratory, carried out a screen of about 3,000 fly lines using this approach at Genelia Farm in collaboration with Jerry Rubin, who generated this vast collection of Gal4 lines that he gave us access to. And after carrying out this screen, we found about 20 hits that tested positive after retesting, and that's about a less than 1% hit rate, which is a little low but not bad for a genetic screen. And interestingly, among our hits, we affected different types of aggressive behaviors. And remember, this is all gain of function. We're looking for manipulations that increase aggressive behaviors, not that decrease them, because there are many uninteresting ways that you can break a complex behavior like aggression. And one of these, what I'll call singlets, that is a hit that affects one behavior, with quotes, is one that causes the fly to exhibit what's been classically described as wing threat behavior. Now these lines typically label 50 or 60 neurons. And so for the biochemists in the audience, what we do in this game is two things. One, we refine, imagine this is enzymology. Where's Tom? We refine our assay, which is behavior, and make it as quantitative as possible. And then we purify the neurons, and we purify the neurons genetically by using intersectional genetics to make the flies with the smallest number of neurons that show the behavior. So Brian Dijkstermars, a postdoc in the lab, carried out a detailed and quantitative analysis of threat behavior in Drosophila, and he found that he could decompose threat behavior into three basic types of action. One, called orientation, where the fly turns to face its opponent. Second, forward charge, where the fly moves towards the opponent. And finally, wing elevation, where the fly puts its wings up in the air, presumably to make itself look bigger. And interestingly, these behaviors, although they are typically exhibited in a characteristic sequence during a threat display, can be combined in a flexible manner, so that some of the threats don't have the wing elevation, but they have the orientation and the forward charge, etc. And through this genetic purification of cell types, Brian was able to isolate flies in which only 3 neurons were labeled on either side of the brain. And amazingly, activation of these 3 neurons can recapitulate in single solitary flies the entire pattern of threat behavior, including the orientation, charging, and the wing elevation. But remarkably, they do this in a scalable, graded manner, reminiscent of what I showed you in the hypothalamus, with at low intensity of stimulation, some behaviors being exhibited, the turning being the lowest threshold, and then the charging and the wing elevation and the pumps requiring the highest level of stimulation. So these and other data, which were just published at the end of last year, if you're interested, suggested the idea that these neurons, again, may comprise some kind of a rheostat-like control that integrates information about the motion of another fly, and also we showed olfactory cues to variably control the nature of threat behavior according to the relative proportions of these different behaviors that are displayed. And perhaps this is a way of synthesizing the views of Lorentz and Tinbergen, the idea that these hierarchical behaviors, this idea may be true to some extent. At least we identify a module that controls threat, and I didn't mention this, but it's very important, it doesn't affect any other aggressive behavior. So if you silence these neurons, you cleanly eliminate threat without affecting lunging, tussling, or any other behavior. But the idea is that perhaps you can merge these two concepts with the view that it is not simply which node that is activated that determines a particular behavior, but that the level of activity in a particular node has some influence on the choice between behaviors at the next level of the hierarchy. Again, a testable hypothesis, but interesting that in both flies and in mice we're seeing similar kinds of phenomena. Now, speaking of hierarchy, what about this idea of Tinbergen's that there's some superordinate node that controls reproductive behaviors, including both mating and aggression in some sort of hierarchical way? Well, the screen that Eric Hupfer performed, interestingly, among the 20 hits, identified 3 independent hits where there was an increase not only in fighting, male-male aggression, but also in male-male courtship. And when Eric looked at the cells that were labeled in each of these 3 lines, and when I say these are 3 independent hits, they're separate Gal4 enhancer lines made from different pieces of DNA. And coincidentally, these neurons, although these lines, although they labeled 50 or 60 neurons per line, there seemed to be a small subset of neurons in each line that appeared to be in common. So we asked the question whether indeed it's the same neurons in each of these 3 hits, and if so, whether those neurons are responsible for one or both of these behaviors. And this is kind of like a complementation test in genetics. The way that we do this is we use a version of the split Gal4 system in vivo. We develop hemidrivers for each of these different lines, one of which expresses the DNA binding domain of Gal4 with a leucine zipper, the other of which expresses the transcriptional activation domain of Gal4 with a leucine zipper, and then we cross them pairwise, and in flies that inherit both of the hemidrivers, we have a GFP reporter, and if there's no commonality between the cells labeled in the 2 parent lines, no cells are labeled in the progeny. But remarkably, in each of the pairwise crosses we did between these 3 independent hits, we were able to get in the intersection the same population of about 7 to 10 neurons labeled on each side of the brain. So genetics works. These are 3 independent hits of the same population of cells, and amazingly, these neurons resembled very strongly a population of interneurons called P1 that were independently identified by Barry Dixon and his group in a similar sort of functional screen for neurons that control singing, courtship behavior. But we confirmed that when we activate these neurons, which are a subset of the 20 neurons per brain that hemibrain that Dixon discovered, we promote both lunging behavior and wing extension. So how can the same population of neurons promote both of these behaviors? Well, these are male-specific neurons. Many, but not all of them, express a master transcription factor that determines male neuronal identity in flies called fruitless. And we speculated, perhaps, as did others, that maybe the fruitless positive neurons, since fruitless is important for courtship, control courtship behavior, and fruitless negative versions of these cells control aggression. But that appears not to be the case, because we can do a triple intersection where we get it down now to 2 or 3 neurons per hemibrain that are the subset of these neurons that express fruitless, and activating those neurons promotes aggression as well. So that indicates that it is within the fruit positive subset that aggression is being promoted, although it still doesn't tell us whether it's the same neurons or different neurons for both types of behavior, and we're still trying to sort that out. However, we got more insight into how these neurons control these social behaviors when we were able to switch from thermogenetic activation of these cells using a trip channel, which takes many minutes to activate the neurons, to optogenetic activation of these neurons using redshifted opsins. And many of you may not realize, particularly if you work on mice, that for the first 10 years of optogenetics, it was not possible to use optogenetics in adult Drosophila because the blue light that is used to activate classic channel rhodopsin doesn't penetrate the fly's cuticle. And so we collaborated with the late Roger Chen, who developed a red-shifted opsin called Reach-R, and Ed Boyden developed a variant called Crimson, and now using these red-shifted opsins, since red light penetrates the fly cuticle, we can finally control the activity of central brain neurons deep in the brain in freely moving flies with millisecond resolution. And what we learned from these experiments is the following. If you activate these P1 neurons in a solitary fly with red light, the fly sticks out its wings and sings, and interestingly, it continues to sing for 2 to 3 minutes after you stop stimulating these neurons. Furthermore, if we do a variant of this expression to look at aggression, where we put 2 of these flies like gladiators on either side of an opaque barrier, we stimulate them for 30 seconds using Crimson or Reach, and then we wait 10 minutes and remove the barrier. These flies will now start fighting with each other, whereas control flies that are not expressing Crimson don't fight, because these are all group-housed flies. So together, these data suggest that activating P1 neurons is producing some sort of a persistent internal state. You could call it social arousal or some other sort of state that increases their likelihood of participating in either courtship behavior or in aggressive behavior. And so we've been very interested in trying to understand how this persistent internal state of social arousal, if you will let me call it that, is controlled. And one of the first things that we learned is that the persistence is not encoded in the P1 neurons themselves. So here's an experiment that we can do in flies where we can both optogenetically stimulate P1 neurons with red light while imaging the same cells using GCamp. And what you see is very transient responses with time constants that are an order of magnitude too fast to explain the persistent wing extension that we observed. So we reasoned that perhaps the persistence is encoded downstream of these P1 neurons in a subcircuit, and we wanted to develop a systematic approach to finding where that persistence is encoded. And so the way that we did this, again, using what you can do easily in flies, and which is more difficult in mice, is an actual, what you could call a functional connectomics screen. So we activate P1 neurons in a fly using a red opsin, and then in the rest of the fly's brain, in about 2,000 neurons, we express GCamp, and we do pseudo 4D imaging through the entire fly's brain every time we activate the P1 neurons. And we identify postsynaptic cells, or candidate follower cells, that are activated in response to P1 stimulation, and we ask whether any of those cells are activated in a persistent way. And indeed, among the 6 example cells that I've shown you here, there were a couple of examples that show persistent activity lasting many, many tens of seconds, whereas there are other cells that show only transient time-locked responses to P1. Now what we can add in is a photoactivatable GFP into these same flies that allows us to essentially fill these neurons. So we're doing stimulation, recording, and filling, all with light, all in the same preparation here. That gives us the morphology of these cells. That allows us to create a search image to find in a GAL4 library a GAL4 driver that labels the population of cells that we labeled here, and to confirm now that when we stimulate the P1 neurons and image from these cells, which we call persistent P1 follower cell 1, they do show a persistent response. Now we have not just look but don't touch, but we have genetic access to these follower cells, and we can ask what is the effect on the fly's behavior of silencing or activating these persistent neurons. And so here's an experiment where we ask whether these neurons are required for the effect of stimulating these cells to cause persistent wing extension. I'm sorry, the wing here is bleached out, but this is the behavioral equivalent of a genetic epistasis experiment. So we combine activation of P1 neurons using a GAL4 driver to drive the expression of the red-shifted opsin with LEX-A-driven expression of a potassium channel that shuts off the persistent P1 follower cells. And interestingly, what you can see here, and again, I'm sorry it's bleached, but you'll see that silencing these PPF1 neurons has no effect on the wing extension that is evoked during the light on, but it largely suppresses the persistent wing extension that is seen after the light is shut off. So these cells are required for persistent but not time-locked activation evoked by stimulating these neurons. And we see something very similar for aggression as well. That is, we suppress the persistent aggression which is evoked by activating P1 neurons. Now, interestingly, stimulating these cells directly has no effect on behavior. So this is a control fly, this is simply wing extension behavior, and this is activating these cells, and here's activating the P1 cells. This is a positive control. And if you think about it, if we believe that these neurons control some kind of internal state, there isn't really a reason why they should produce a behavior. Maybe all they do is alter the behavior the animal shows in response to a stimulus. And so we tested that by doing the stimulation in the presence of a dead female. And when we stimulate these PPF neurons in the presence of a dead female, we get a much stronger singing response to the dead female than we just get with the dead female alone without stimulating it. This is important because this is a criteria that these neurons have an influence on internal state. They're changing the input-output relationship between the sensory stimulus and the behavioral output. Now, what about physiologically? As I told you, these neurons are persistently activated by stimulating P1 neurons, but if you stimulate these cells, the persistent follower cells directly, they don't show any persistent activity on their own. So that raises two possibilities. One is that these neurons don't actually encode persistent activity. They are simply inheriting it from some upstream persistent center. The other possibility is that they are part of a persistent circuit, but activating those cells alone is not sufficient to trigger persistence because other cells in the network have to come along for the ride. So the experiment we want to do to distinguish these is to ask if we break activity in these cells during the persistent phase, but break it only transiently, does the activity come back after we stop the breakage? If they're just inheriting persistence from someplace upstream, then as soon as we relieve the brake on activity, the cell should rebound to the normal level. On the other hand, if we silence these cells during persistence, and then after silencing, they don't rebound, that suggests they're part of the mechanism. So to do this experiment, and again, these are new and relatively unpublished data, we combine now stimulating these cells with imaging in either the PPF population or another persistent population, and we silence these same cells while they're in the persistent phase. So what you can see here, I'll take you through this, we stimulate the P1 neurons. This is the trace of imaging the activity in the PPF1 cells with no perturbation. Now here's the phase during which we silence the cells briefly in green, and you'll see that after we turn off the silencing, the activity pretty much does not rebound to where it should have been, whereas if we do this in this other PPF2 population, after the silencing, then the activity rebounds back to where it would be according to this persistent slow decay. So these data, which are quantified here, suggest that these cells are part of the mechanism that encodes persistent activity, and they're not just inheriting it passively from some upstream center, but it also suggests that there are other components besides these cells that need to be recruited in order to produce persistence. Otherwise, stimulating these cells by themselves would have yielded persistent activity. So what these data are suggesting is that activating P1 neurons triggers two different kinds of pathways, something that I'll call a command pathway, which promotes overt singing behavior, and then some sort of an internal state pathway, which is persistent, which facilitates the animal's engagement in either aggressive behavior if it encounters a male, or in courtship if it encounters a female. And specifically, we know that these P1 cells, from work of Vanessa Ruda, Kristen Scott, and others, these P1 neurons are activated by female pheromones. So the idea is that maybe following an encounter with a female, when these P1 neurons are active, this persistence mechanism allows the male to walk around on his leaf carrying a persistent memory of the female, so that if he then encounters another male, that he will combine that information with the memory of the female to trigger aggression. Because flies, like people, don't fight unless they have something to fight over. They have to have a female to fight over, food to fight over, or territory to fight over. And so what I've shown you in this part is that P1 neurons trigger a persistent internal state that promotes social behavior. The persistence is encoded downstream of the P1 neurons, not in the P1 themselves. These cells, follower cells, encode some sort of internal state, not a command function, and that this could reflect a persistent memory of a female. Now, in just the last couple of minutes, I'd like to quickly go over a recent story that addresses the question of whether there might be conserved genes underlying aggressive internal states, where we moved from our work in flies to our work in mice. And specifically, we initiated a study in flies to try to identify neuropeptide-containing neurons that promote aggression, because work from a number of laboratories, including Kurt Bargmann and Ulrike Heberlein and others, have shown that there are specific neuropeptides that play evolutionarily conserved roles in survival behaviors like feeding and pain. And so we carried out a small-scale, unbiased screen to activate fly neurons that express each of the 25 neuropeptides that we knew about at the time that we initiated this study, and measure whether activating any of those cells promotes aggression. And what we found, surprisingly, is that only activating flies that expressed the relevant ion channel in tachykinin neuropeptide expressing neurons caused an increase in aggression, and we were able to show that the peptides themselves are required for this behavior, and that the tachykinin receptor is required for this behavior as well. And we think that this peptide increases some sort of aggressive-like arousal state, because if we activate, if we activate these neurons in a fly exposed to a moving magnet, which is about the size of a fly but has no olfactory or any other cues that make it look fly-like, we can occasionally get the flies to attack the magnet, which they would ordinarily never do. And this is a little bit like the fly version of the famous Tinbergen experiment, where he showed that when a male stickleback fish were in their high arousal, fighting and mating season, they could attack dummies, that some of which look hardly anything like fish, as long as they had an eye spot and a red belly. So, interestingly, in flies, our data show that this peptide seems to play a role in the control of aggression by social isolation. So flies, like many other animals, only fight if they are socially isolated. In group house flies, aggression is completely suppressed. And we showed that a mutation in this tachykinin peptide gene makes socially isolated flies non-aggressive, like group house flies, and that conversely, overexpressing and hyperactivating tachykinin neurons in group house flies makes these flies behave as if they were single housed. And most recently, we've confirmed that single housing itself upregulates expression of the tachykinin gene in the male fly brain. So we were interested to know whether anything like this could be going on in mice. And in mice, as in flies, social isolation is, and I'm talking about 2 weeks of social isolation, is well known to increase aggression. So these are group housed mice, socially isolated mice. And Muriel Zelikovsky, a postdoc in the lab who's now going to run her own lab at the University of Utah, made the remarkable discovery that after 2 weeks of social isolation, the levels of tachykinin 2 peptide expression in the brain are increased so strongly, in this case using a GFP reporter, that you can tell on a benchtop which brains came from isolated mice versus group house mice, because the brains from the isolated mice are green. And so this raised the question in our minds of whether there was any causal relationship between the upregulation of tachykinin in the mouse brain and the behaviors like aggression that are produced by that isolation. And in addition, I should say, to increased aggression, the effect of social isolation also changes many other behaviors, including defensive behaviors. It converts stimulus locked freezing to innately aversive and conditioned fear stimuli into persistent responses. So initially, we did a loss of function experiment. We took advantage of a drug called osanetant, which is a specific antagonist to the NK3 receptor, which is thought to be the receptor for tachykinin 2, whose protein product is called neurokinin B. And remarkably, injection of animals systemically with this antagonist completely abolishes the effect of social isolation to produce aggression. Furthermore, we found that treatment of the animals with this drug is protective. If we inject the animals with osanetant before social isolation and isolate them for 2 days and inject them several more times, they don't fight. Conversely, and I should say that the antagonist also blocked all of the other effects of social isolation, persistent freezing, to the looming disk, to the conditioned stimulus. And so we did a gain-of-function experiment, and this is a novel experiment, a sort of first-in-class experiment we did with help from Viviana Grabnaru in the lab, and again, it took off directly from our fly experiments. What we did here was to combine overexpression of the tachykinin neuropeptide in group house mice with chemogenetic activation of tachykinin-expressing neurons, reasoning that if we only overexpress the peptide, it's going to be like filling up a water balloon. You've got to do something to make them release it. And only if we combine those two manipulations together in group housed mice, we get increased aggression. We don't see this if we only overexpress the peptide without activating the neurons, or if we activate the neurons without overexpressing the peptide. And I was absolutely amazed that this experiment worked at all. I can't say that this manipulation affects all of the behaviors produced by social isolation equally, but it makes a strong case that the upregulation of tachykinin is causal to the increase in aggression. We also addressed the question of whether tachykinin affects all these different types of behaviors, aggression, persistent fear, by acting in a hierarchical way from one central hub in the brain that acts on many downstream structures, or which indirectly affects such a hub, or whether it acts in a distributed manner to control different behaviors in different brain regions. And we know that it is upregulated in many different structures, and I won't go through this in detail, but we did this ghastly multiplex experiment where we interrogated 3 different brain regions with 3 different types of manipulations and tested them in 4 different assays. And the bottom line is that this peptide affects multiple behaviors by acting in a distributed manner. It is upregulated in parallel in different brain regions, some of which control aggression, like the dorsomedial hypothalamus, some of which control acute freezing, like the central amygdala, and the other of which controls persistent freezing, like the BNST. So this is an example of a state, an internal state produced by social isolation that you can think of as being cobbled together from many independent sites of upregulation of this peptide. So does this have any relevance to people? Social isolation is clearly a major source of mental illness, or it exacerbates mental illness. There have been an increasing number of articles in the lay press about this, but I think not enough consideration given to the issue of how social isolation stress affects the brain. If nothing else, these studies show that social isolation can profoundly alter the chemistry of the brain in a way that has lingering effects and that can influence multiple different behavioral domains. Interestingly, osanetant has been tested in humans as a treatment for psychiatric disorders, but in virtually all of those tests, it failed due to lack of activity. It was primarily tested in schizophrenia and panic disorder and major depressive disorder. So osanetant, which was well tolerated by subjects in phase 1 clinical trials, now lies on the slag heap of abandoned drugs which the pharmaceutical industry tried once or twice and abandon and move forward on, but these observations raise the possibility that perhaps this drug or drugs in this class might indeed find a useful therapeutic indication, but not in the symptom domains that were originally chosen to test it for efficacy. So to summarize, I've given you 3 stories about mechanisms, different types of mechanisms that underlie persistent internal states associated with emotional behavior. One involves a glutamatergic circuit node in the hypothalamus that controls a persistent internal fear-like state. Another one involving a relay in flies that controls a persistent internal state of social arousal. And finally, a neuropeptide that's upregulated by social isolation that enhances aggression and converts stimulus locked to persistent defensive responses. And I just want to stress that these kinds of studies, I think, highlight the importance of comparative approaches in studying multiple types of model organisms in trying to ferret out general principles of brain function. And despite the fact that ultimately we want all of this to impact humans and our health and well-being, we need to make sure that we continue to exploit the power of genetic invertebrate model organisms as well as vertebrate mammalian systems as well. And with that, I'll thank the talented students and postdocs in the lab. This was the cell cover we didn't get that have contributed to this work over the years, and to thank you for your attention. I'll be happy to take questions.